Yeah, so this workshop is going to be kind of focused on some of the methods that are being developed to try and standardize how we visualize our synthetic biology designs. Originally, this was not just going to be me presenting this. It was also James Scott Brown, who also is known as one of the editors for SBOL. And unfortunately, he's in Scotland enjoying a nice break. So he's been able to record a short video that kind of talks through some of the aspects that he was going to speak about. So there'll still be an aspect of that, and I can answer any questions you have about what he's going to talk about. But like I said, this is quite informal, and if you're interested in coding and potentially actually trying to use some Python code to produce these diagrams programmably, then there's also a GitHub repository that I've put together. So this link down here. What I'll do actually, I'll copy this into the chat. So if you want to go there, you're welcome to clone that. And oh, Prashant's already done it. Wow, he's ahead of the curve. Brilliant. So yeah, at some point there'll be some coding bits I can talk you through, but that's there with a lot of the information on it. So in terms of structure for today, it's going to be basically broken up into 3 bits, and it might be that you want to attend a portion of this and other bits aren't of much interest to you, but I just want to go through kind of overview of what's going to be covered. So to begin with, I'm just going to give a very brief introduction to what SBOL Visual actually is, some of the kind of rationale behind why it was developed and how it works. And I'll also end by telling you a little bit about some of the kind of future directions that have been discussed within the community and ways that you can become involved if you're interested in contributing. And then the second part is going to be, first of all, a presentation from James, which is around some of these future directions for SBOL Visual. And this specifically focuses on a new kind of file format that we're trying to promote for actually describing the symbols that we use within these diagrams, which is based around a format called Parametric Scalable Vector Graphics, or SVG. And he's going to tell you a little bit about why we care about that and how we've been trying to integrate that into the standard. And then to end, I'm going to do a bit of live coding, which I could crash and burn. We'll see what happens. But I think it's good for you to see how easy it is potentially to create diagrams using some code, which is really valuable when you're starting to look at kind of automating some of these processes, or you've got very big libraries that there's no way that you could manually sort of like create diagrams for. So will hopefully give you an idea of a really simple library that can give you access to some of the symbols that are necessary in order to create these standard compliant diagrams. So to begin with, I just wanted to highlight kind of a problem, and kind of this is kind of the rationale behind why we started to create SBOL Visual. And that's the fact that when you look in the literature, and this is probably from like maybe even 10 or 15 years ago, you find that there's a real mixture of ways that DNA or genetic constructs are visualized. So this is some examples from some ACS synthetic biology papers, and you can see that you've got sort of some elements that I'm assuming these are genes, sort of represented by these arrows. We've also got these arrows, in this case it is representing what looks like a T7 promoter. Here you've got bars, and here you've got some of these diamonds. And so there are lots of different symbols here. And in many cases, what you find is that some are similar, but some are different, and some seem to have conflicting meaning between what they're actually used for. And although this might seem like quite a trivial point, it's these visualizations of these designs are really important for us understanding what's actually going on potentially. And so they can actually make it very difficult for us to be able to understand what someone's trying to describe when they're actually showing us their design that they actually want to build. And so to try and get around some of those problems with actually being able to communicate kind of our genetic designs, we took kind of inspiration from other engineering fields, such as electronic engineering, where for a long time they've kind of taken the elements that make up their circuits in this case, and they've kind of codified in a graphical form what each of those elements should look like when it's actually visualized. And so this standardization effort makes it much simpler to actually allow two electrical engineers to look at the same schematic of a circuit and actually understand, firstly, what each of the elements are doing and how they're connected. And that propagates through to actually better understanding what their actual circuit or design might be doing. But like I said, we didn't previously have an equivalent of this kind of set of symbols that we can use for synthetic biology. And the idea behind SBOL Visual was to really see if we could develop a community standard that would at least agree on how certain types of genetic element or components and their interaction should actually be represented. Now, to kind of give you an idea of what this looks like, so some of these symbols and diagrams you'll have seen if you've been to any other talks and the conferences. In this conference, you'll likely have seen some of these symbols already, although they may still have been used in a slightly different manner. And so the way that a genetic design is represented in SBOL Visual is by, first of all, having what's called basically a backbone. So that's just represented by this kind of line here, which is effectively the DNA that we're representing. And then on this line, we basically add symbols which correspond to different types of part that are actually encoded within the DNA. And so these symbols allow us to basically specify the ordering of different types of parts, as well as the orientation. So you can see this arrow here is pointing to the right. So this is a promoter that's on the actual, the kind of the forward strand of the DNA, whereas this one's pointing to the left, so it's going in the opposite direction. And so the symbols that have been chosen for a lot of these elements are specifically set up so that you can very easily see their orientation, either by flipping them to the opposite strand or by kind of the shape actually is non-symmetric along this axis, so you can tell which direction it's actually encoded within. And so, like I said, you can use this to then capture a number of different sequence features, which are shown by these different symbols. As I said, you can have reverse complement versions of those. These can then be labeled. So if you want to help a viewer understand what exactly is this promoter, okay, it's the p-tet promoter. That's always helpful. But one of the things which SBOL Visual does that goes beyond just looking at the DNA itself, it also is able to capture interactions between those genetic parts. So this includes things like, say, regulatory interactions. So here you've got this symbol basically means it encodes for a gene. And so this gene is going to repress, in this sense, this promoter here. There can be additional small molecules that are interacting with some of these interactions. So in this case, this small molecule ATC is actually having a negative effect on this repression of p-tet. And we can also represent things like molecular species that might be synthesized from the DNA itself. So in this case, if we've got this GFP gene here, it would be transcribed into an RNA or a transcript, and that's what this little squiggly symbol represents. And then this squiggly RNA would be translated into a protein. And so we can capture these additional species, which aren't just the DNA itself, through another set of symbols that we actually have access to. And here we can show that a process is taking place by using basically arrows. Another thing that's really a nice element of SBOL Visual is that as these designs get bigger, they just become very, very complicated, and it can be difficult to know what's doing what. And so there's also the ability to add effectively a hierarchical structure to them by kind of encoding elements within what we would call a module. So this whole element here would be thought of as a module, and it helps the viewer then understand which parts of the genetic design are corresponding to maybe a specific function. And as I'll show later, you can potentially simplify these modules into kind of abstract forms. So you don't necessarily then have to include all of these genetic details. You just know that this is a module that has one input and one output. And so you can then abstract away some of the details of the genetic design to focus on just showing the elements that are most relevant for what you're trying to get across in terms of to a reader or someone who's actually going to make use of this design. So that's kind of a basic overview of the kind of the main components that you'll see in an SBOL visual diagram. But one of the key elements of the specification is that we don't actually kind of force a very rigid style on you as a user. We're open to allowing symbols to be drawn in a number of ways. So we're very flexible in terms of like the colors that you use, and as long as the shapes are representative of those that are found in the standard, that's also completely fine. You can make things bigger or smaller. You can, like I said, change their colors or their line widths, all those sorts of things. And the reason for having this flexibility is that we don't want this standard to just be something that you would use, say, on a computer. It's designed to try and be easy to use by anyone, even on, like, for example, a whiteboard. So a lot of the symbols are designed to be as simple as they can, but different enough such that if you want to scribble something on a whiteboard quickly, you can, and it's understandable, and it's easy to actually see what's going on. So there's a lot of flexibility there if you want to use SBOL Visual. In terms of the symbols that are available, I'll show you in a minute a website where we actually define the standard, and we have what we call samplers, which basically show all of the symbols that we have ratified in the standard. But this is just a very brief overview of the main glyphs that are currently present, and the ones that you're probably most likely to see in diagrams if you looked in papers in the synthetic biology literature. So I'm not going to go through all of these because there is a lot here. But things that are very commonly used are things like, for example, coding regions. You might have seen some of these, like you may have kind of a polypeptide region where several elements of a gene are kind of encoded. This is a new part, actually, in Intron, to show sort of a separation of things. Promoters you will have definitely seen at some point, I suspect, through the conference, and things like terminators as well. And one of the nice aspects about these nucleic acid glyphs is that we can also capture things like that happen perhaps at the ends of a DNA molecule. So, for example, we can also represent different types of sticky end, which might be useful if we're wanting to demonstrate maybe a type of assembly that we've come up with. And we can also abstract away details, as I said, by making things modules, or by just saying that we have these specific things happening in a design, and then there's another region which is also engineered, but I'm not going to show the details of that. And so we have kind of shapes that represent these things. And we can also kind of zoom in and zoom out of these elements by using other types of symbol on the backbone itself. Now, all of these are kind of actual things that were being coded like within the DNA. Often you also, in a design, you might want to specify specific locations that are important. That could include things like where this design is actually located in the chromosome of the organism. And so we have these kind of like S-like shapes that you can place at the end of your design, and then you can include a locus or whatever that to actually define where that design would be found within the host itself. Or if it's a plasmid that you're actually describing, you can use these little curved ends to kind of show that it's a plasmid that's actually encoding this design. And similarly, within a design, you might have specific locations that you want to highlight or that have some functional relevance. And so we have these little bar-like shapes to represent DNA positions. If a position is relevant to the RNA that's maybe synthesized from the DNA, you can use this kind of squiggly little line. And if it's actually related to the protein that's encoded by DNA, you can use this kind of spring-like shape. And one aspect of these locations which is quite nice is that they can also capture kind of functional behaviors that might occur at that particular point. So for example, stop codons, stop codons in say an RNA, or maybe a transcription end site, transcription termination site even, as well as things like cleavage sites, or perhaps even sites that kind of promote stability of the element itself. Now, the only other thing I want to say about these is that when you look in the standard, what you'll find is that there are recommendations for the symbols that you should use, but sometimes there are alternate versions. So the most obvious example of that is the actual CDS. So this is the symbol that we would recommend that you use for showing CDSs, but an alternative that we also say is okay is this arrow-like shape. And the reason that you'll find these kind of alternative versions is that historically, like any standard, things get added when enough people use them, but before then, people sometimes have different ways that they actually represent the same things. And so what we've found as the standards evolved is that we move more and more to a single symbol being used for a specific type of part, but there's sometimes it takes a bit of time in order for that to happen. And so that's why these alternatives are sometimes present. So those are some of the actual elements that can be encoded in the nucleic acid itself. We also have some kind of more general symbols for molecular species and kind of the interactions between the DNA, RNA, and proteins within your design. And so we have things like, for example, if you've got, say, a complex that forms, we allow, for example, a superposition, where you can superimpose those two symbols of the things that are coming together to kind of represent that complex. We have some other kind of rounded rectangles for macromolecules, this kind of pill-like shape for proteins. And then there's a number of other different shapes for kind of molecular species that might be of use. And then, as I said, we also have these different types of interaction, which are just represented by lines, to capture things like some kind of element of control that's taking place. A common one is degradation, if you're looking at kind of different ways that something's being expressed. And you've got things like inhibition, process, which might mean something's happening, and then stimulation, like, for example, activation of a gene. And in addition to these interactions, we also have these, what I'd call kind of, I'm not sure what you call these actually, but you may have a situation where multiple interactions come together. So for example, you might have, for example, two proteins that need to associate to form some functional macromolecule complex. And so these symbols here allow you to more precisely say what's actually happening when these two things are interacting. And so you have things like association, dissociation, or processes. In a lot of cases, there are also these kind of unspecified things that you can include if you're not really, if it's not unknown what's actually going on. But I've never seen a diagram actually use these, because normally unknown things people will either completely ignore or not include in their visualizations themselves. So with these two sets of glyphs, you can pretty much describe most of the sorts of designs that you might need to in synthetic biology. And so they're a real kind of, they're really useful for actually building diagrams where the actual elements have an understood meaning, and other people within the field can actually make sense of them. So I've got to stop there. So I wanted to ask, does anyone have any questions on these symbols and how they're used, or more generally about how SBOL Visual works? Or does everyone want me to carry on into actually showing you how to make some of these diagrams? I can't do a vote here, so you'll have to either put something in the chat, or if you'll stay quiet, I'll just carry on. Okay, so I've got one from Alex. A complex or a macromolecule? So I would say, yeah, that's a difficult one actually. It could be both, I guess, and you could represent it by both. I've tended to find, so the question was, would a protein dimer be a complex or a macromolecule? Oh, let me go back. And so I've tended to see in diagrams that people would superimpose, say, several proteins together for, say, a dimer, or some other number of elements that need to come together, because it actually it shows you what's actually happening, I think, a little bit more visually, which I think actually is a bit easier to understand. But it doesn't mean that the other way wouldn't be acceptable. It's just whatever makes most sense in terms of getting across what you're actually showing. Michael's asked, could you say how colors, text labels, and keys legends works? Yeah, so there are no guidelines. So I showed you this thing here before. There are no actual guidelines in terms of what colors should mean or any styling element should mean at all. And the reason for that was that often the style of the glyph that you use, your diagram's drawn to kind of basically communicate a very specific thought or idea. And so we wouldn't want to kind of force the use of colors in a specific way for only a specific meaning. And so we allow you to use them however you would want. And it's really up to you to think about how best colors and styling would actually convey the actual point you're trying to make in a diagram. But normally what you tend to find is that colors are used to, for example, show, this isn't a good example here, I can show you one later, but you might, for example, have a repressor or a transcription factor which interacts with a promoter. And so often color would be used to show that this transcription factor interacts with this promoter, so they'd have the same color effectively. People do sometimes write the names of promoters and genes, but sometimes those interactions aren't always that clear. And so just by using color, it becomes very evident that those two things are similar, or they interact, or they have some kind of functional link that we're not sure that you want to kind of get across. Any other questions? No. All right. Well, if you think of any others, do feel free, just keep putting them in the chat, and I'll try and address them when I get to the question bits. But yeah, this is basically the library of elements you have, and you can put them together in this way, and you can make designs that most people can understand, which is good. Right. Making SBOL visual diagrams. So there are lots of different ways that you can potentially actually get started to create these diagrams. As I said, you can use these with a pen and paper. There's no problem doing that. But obviously, it's probably not the most efficient way to create kind of more complex diagrams. And so if you want to have kind of basically these glyphs in a format that you can use in, for example, some graphic design software, we actually provide, let me stop sharing now. So can everyone still see my screen? Yeah. Oh no. Oh no, wait a minute. I didn't want to stop sharing my screen. I always do that. So can everyone see my desktop now?
Yeah.
Yeah, all good, cool. Okay, so if you're wanting to create some diagrams on the, let me see if I can find that. There's a website called SBOLstandard.org. And this is basically the home of all of the information about both the SBOL visual standard and also the data model, which is another standard which can be used for kind of representing representing the kind of a biological design on a computer. But on this website, if you go into the visual part, you can find out more about SBOL visual. But the most important thing if you want to make some diagrams is there's this download glyph section here. And so you can click on this little link here, and this is basically the samplers that I told you about previously. So these are actually automatically generated from a Github repository, which we use to actually manage the standard from. And so this shows all of the glyphs that have been ratified, and it's possible to go into the standard and actually find out what this actually represents and means and all the rest of it. But when you're creating some diagrams, you just need files that encode each of these. And so if you go to the releases page, you'll be able to find the latest release, and there are this number of different glyphs sets, basically, either in a PDF format, a PNG format, or SVG format. Now, I recommend if you want to say create a diagram using, I don't know, PowerPoint, and I'll show that now. The easiest ones to use these SVG formats, because you can actually, PowerPoint, a lot of graphic design software understands SVGs, and so you can then start to edit the style of the actual symbols very, very simply. So I've downloaded this previously, and so where is it? And so this is what you would end up having. You'd have this folder of all these different SVG files, and each of these corresponds to a particular symbol that you might want to use. Now, to show you how you can use this in something like PowerPoint, so this is just a slide where I've literally drawn a line on it. And what you can do is you can then, for example, if we wanted to make a really simple construct, we can look through here and say, okay, I'd like to start, I'll make the stereotypical kind of transcriptional unit. So I want a promoter. So you can literally just drag these in, and you can see these are then little elements, and you can resize them. But the really nice thing is that you can also, because they're kind of vector based, you can actually change things like, for example, the thickness of the line. If you can see it's getting thicker, maybe you can't see that. The thickness, you can change the color of it, for example. You can do all sorts of things that would be quite hard to do if it was just an image. They're actually editable. We can add some more things in here. So let's add a normal CDS. And so with this one, it's a bit bigger. This one we can also fill it in because it's actually it has some color. So let's make an orange one of these, and then let's have another terminator. Put that here, and then we can have another. Let's have another promoter. So make the well-loved not gate type regulatory link. What I'm going to do is I'm going to make this actually the same. We were talking about colors earlier, so I'm going to make this orange. So maybe there's some association here. And the really nice thing about kind of using something like PowerPoint or any design software, to be honest, is that you can often they have functionality to actually connect together things. So you can, for example, use these connectors to actually connect different elements, and that's potentially really useful if you want to say this maybe activates this. End arrow type. So we can say, let's say it's an activation, and make it a bit bigger. It's actually very, very small. So it really gives you a way of using these symbols very quickly to make kind of diagrams that are then standard compliant, and hopefully people will better understand what you're trying to convey. The other nice thing about using SVGs, and this is where I am going to potentially crash and burn, is what I believe you can do is you can actually convert this to a shape. I think it's this one. And then you end up having each of the points within the symbol as editable. So you can then potentially even, you might, for example, want to make this a more arrow-like one. And so these SVGs are really good for being able to start from something which is kind of standard compliant and tweak it in terms of style and shapes and things to get it just how you want. And that tends to work really, really well. Another piece, so PowerPoint's fine for doing these sorts of things. Another piece of software I use a lot is Affinity Designer. That's another kind of a Adobe Illustrator style piece of work, and these work really well in that as well. So these SVG files are really great for being able to make your own diagrams very, very quickly. Okay, I won't say that. So that's one thing you can do. I've shown you where to get the glyphs from. In addition to kind of making these diagrams manually, there are, if you're a developer, for example, maybe you're helping to, I don't know, build a website that's visualizing, I don't know, a library of metabolic pathways or something like that. There are a lot of tools to actually help, especially with web-based tools to visualize using SBOL Visual. And so one of them is called SBOL, which I think I've got here. And what that allows you to do, I'm not a web developer, but it has components that you can incorporate into your website. And they will actually then allow you to render SBOL designs correctly and accurately, and basically handle a lot of the the effort of actually drawing these diagrams for you, which is really, really useful. So yeah, have a look at SBOL, or sorry, VisBOL 2 if that's something that you're interested in. There are also, one of the things that's been emerging is actually web tools for making these diagrams. And so this is something that's kind of Chris Meyer's group, which is called SBOL Canvas. And so this is a kind of a, you can imagine like an online design tool for making constructs. And this is also very easy to use. So if we want to make something similar to what I made before, we can just click on these things. I want a promoter, CDS, a terminator, and maybe I want another backbone. So let's put this backbone over here, and this one's going to have a different promoter on it. And I want one of these, I think. And this will let you connect up these things as well. So this tool is really nice for kind of having an interactive web-based thing. I think it has nice features like the arrows follow things and stuff like that. And on here as well, I think there are things you can do in terms of changing the, for example, the color of these parts. Let's make the outline blue or whatever. And so this is a kind of an interactive sort of design tool that you can use. And then from this, you can export to images or other, I think you can even export to SBOL data files, which is quite handy for different things. But it's just another tool which kind of simplifies sort of the creation of some of these diagrams. And then the final tool I wanted to kind of show you is something called DNA Plot Lib. So if any of you are Python programmers, this is a tool that I wrote a long time ago now, basically because I was fed up of having to manually create these diagrams for big libraries. And so with some help from a couple other people, Brian Durr and Emerson, we created a Python library that allows you to, in Python code, effectively define constructs and then render them to matplotlib figures. And I use this a lot. So if you've seen it in my talks, a lot of the diagrams that are shown in it are created through Python scripts. And so this is I'm not sure if is this visible to everyone, or is it too small? Is it a bit bigger? Does that help? Is that visible now? Yeah, I'm assuming yes.
Yeah, that's better now.
Is that better?
Yeah.
Try and make it a bit, I can make it a bit bigger. That's probably about as big before I get struggle moving around. But so you can see here that we can implement sort of complex circuits and designs with regulatory interactions, but these can actually also be embedded within kind of normal figures or graphs or plots that we might be creating. So this is actually a standard bar chart in matplotlib, and these are just appended to the actual design. We can append, for example, a scatter graph. And one of the really nice things about having a programmable interface to create these diagrams is you can start to associate genetic design elements with actual experimental data. So some of these things down here, so for example, this diagram here, you can see we've got a genetic design here, which is actually, it's being represented in a realistic manner. So the length of these arrows actually is the length of the coding regions. And this under here is actually some sequencing data and some inferred model data that allows you to kind of then really see, okay, at this point, what's happening with the read depth, or okay, at this point, we seem to have a high RBS score, and actually, yes, there is an RBS there. So there's ways of associating the data then with the experimental designs, sorry, the genetic designs. And the other thing that I think is really becoming more and more valuable with a lot of these visualization tools, especially the programmable ones, is the ability to create dynamic visualizations, which I think is really important for some of these processes. And so this is just an example. I've got the video here that's output from the script, where we have a genetic design down here at the bottom, and what we're showing at the top is basically an ODE simulation of what we think this design is actually doing. And you can see that the regulatory links and the colors of these things are changing in intensity and size based upon the actual state of this dynamical system that we're using to actually model it. But you could imagine that this could also be updated based on experimental data that you might be collecting as well. So there's a a really nice way of beginning to create visualizations that show the design information, but in the context of perhaps how it's actually functioning in a cell or cell-free system or whatever it is you're using it in. And this is really valuable. And just to show this isn't, these sorts of things are not crazy to code. So if any of you are like thinking, oh, that looks really complicated to do, for example, the code to create this thing, if I make this a bit smaller, isn't crazy. There's some elements at the top which are basically defining what the parts are in the design. Here you've got the actual, effectively the differential equations that govern how the system works. There's a little bit here that actually plots it, such as for Matplotlib, and then most of this is just setting up basically a figure in Matplotlib, which is the right size and has the right things. So the actual amount of code is maybe, I don't know, 50 lines of code of actual specific stuff for the visualization you want. So with very little code, you can produce some really nice diagrams that potentially can be really valuable in kind of explaining what your designs are actually doing or how they're actually functioning. Okay, so that was the last of those specialized visualization tools. Was there any questions on that in particular? So let me have a look through. Oh, thanks Prashant for putting all the links in. Yeah, Alex is right. So PowerPoint, one of the annoying things, this is an annoying thing in PowerPoint. I've even raised this with Microsoft, but I think it's one of those things that not many people use it. In a lot of genetic diagrams, we have this kind of inhibition link, which has this bar at the end of the thing, and that isn't actually present. So actually I can show you this link here. That's not present in PowerPoint. There isn't an end bar like that. So that's one of the, I guess, the annoyances of using PowerPoint. But if you use any other graphic design software, it's almost always present. So I've never had any problems in other things that I've used. So yeah, you have to manually draw those on, I'm afraid. Okay, so there's another one here. A few moments ago, we were discussing with James about including this SBOL2 construct visualization in one of our tools. We tried using it and it worked fine, but what was shown was the whole backbone along the page. Not much visualization. Is it finished now? So I'm not involved with the SBOL project, so I don't know if you're having difficulties with that. It might be worth contacting either James Scott Brown, who would be here for this normally, or I'm trying to think who else was. I think there's the group at Newcastle that have developed a lot of these kind of web-based tools. They might be able to sort of help you out. But I think the tool is complete, but it might just be that you have to use some of the additional functionality that's within it if you need multiple backbones. And I know it will do that because I've seen examples where that's possible. So I don't think it's a limitation of the tool. I think it's just that you may have to, your code may have to actually implement some of that functionality itself. And that's it in terms of questions. Are there any other questions on this this aspect? No. Okay. Well then, for the next bit, I was just going to show some examples. Basically, this might spark some discussion, but of some maybe slightly more complex diagrams, sort of highlight some of the kind of elements that I spoke about previously. So let me put this back to full screen, and yeah, keep adding questions if you have any into the chat. I have that open on the screen, which is good. So yeah, this is just I'm just going to go through some examples of diagrams that might help you understand some of the ways you might represent them with SBOL Visual. And I should highlight that there are often many different ways you could represent the same design, and how much granularity in terms of the detail you want to include really depends on what you're actually using the diagram to show, I think, half the time. So in this case, this was an example of a CRISPR-based device, or a CRISPR-based NOR gate, where you have this promoter and it has these two sites or operators upstream of it, which you can have basically a dCas9 with a specific guide that's complementary for this actually bind. And so you have effectively this complex of dCas9 with the blue guide interacting with this element, and dcast9 with the green guide interacting with this element. And these interactions act as basically inputs to this device, and then the output itself is also a guide, which could then perhaps do some regulation later on. And this is just an example of showing how you can have this kind of complex formation where you've got two symbols here that have basically been overlaid to kind of represent them coming together in some way. And here you can also see the use of this solid circle, which is specifically means that this is a association. These things are actually becoming, connecting in some way. And this is a process. So this is transcription of this. This is basically a non-coding RNA, which gets produced. And finally, like I said, we've got this dotted line. So you can think of this as a module that you might use in multiple cases elsewhere in other designs. And that's precisely kind of what this is kind of representing in terms of you maybe take this module and you can then put many of them together to actually create a more complex circuit and show how that works. And that's really good when you have these very complicated designs with lots and lots of parts, because being able to show which bits are doing what functionally actually helps a lot with the actual understanding. So a question here, which tool set did you use to create this CRISPR diagram? So I think Jake Bill created this one actually, and this was just created using PowerPoint. So I can tell that because you can see these bars are a bit skewy in some places. So they've just been drawn on separately, but all of these are kind of normal links, and that's how that's been created. Okay, so that's kind of one thing. Another thing that I think is quite nice in a lot of these diagrams is, like I said, this isn't a formal specification in a lot of ways. It's often guidelines for how you can kind of make your diagrams more understandable, but it doesn't mean you can often supplement them in nice ways to really help someone understand what's going on in your specific biological system. And so I like these examples because these kind of add kind of some sort of, chemical structures to the diagrams themselves, which is perfectly fine to do, to really help someone understand exactly what it is that's interacting with this ZAR repressor in this case to actually make it active. And this really can, if you're a chemist, this likely will actually help in a lot of ways, understanding where certain chemicals are interacting and what they're doing. Now, when I showed that module on the previous page with the dotted line, one of the things you can do is when you have one of those modules, if you give that a name, it's like a NOR gate, or in this case, an upper pathway, you can then actually represent those as these boxes with these little kind of what we call ports that are on the outsides. And so this allows us to kind of abstract away perhaps a massive part of our system so that we can just show the bit that we're interested in. And we know that this is then going to output into this complicated pathway, but we're not interested in that right now. And so we just completely simplify it as just this black box that we don't know what's going on inside in terms of this diagram. And that can really, if you use this effectively, it means you can often really help a reader understand precisely what it is you're trying to communicate, and they don't get confused by all this other stuff that's going on, which isn't actually that relevant for the point you're trying to get across. And you can see that down here as well, where in this case, this kind of meta box has been actually expanded internally, so you can actually see how it's actually functioning. And this also shows this. So I showed you these kind of expansion kind of symbol. So you can have an element which you can then expand out to sort of show that this actually represents something more complicated internally. Okay, and then the final example I just wanted to go through is around some of these locations. So you'll find more and more, I think, as we probably start engineering more complex organisms, that actually where we're putting things perhaps inside them is becoming more important. And so this is just a diagram where we've used kind of the standard chromosome visualization that's often used. In this case, it's for yeast. And we're trying to show that we've engineered certain portions of different chromosomes by these little red bits. But the nice thing we can do in SBOL Visual is we can use these S-type shapes with the actual locus, so we can really specifically show where this piece of genetic code that we've designed actually gets inserted. And that's really nice because it gives you a nice overview of what's happening, but it also allows you to then really understand in much more detail exactly where these things are actually being placed and what they might be interacting with. This is also a really nice example, I think, of how, although I think the styling is maybe not the most easy to read sometimes, but it's an example showing you how you can also use styling to really get across useful information in a complex diagram. So here you can see you've got kind of normal shaded genes, which are just the kind of the wild type genes, the ones taken from nature. And then there are some which are codon optimized. So you can see these kind of striped white genes. And then you have these polka dot genes, which have also been modified. So it helps you really understand which bits are maybe completely synthetically coded and which bits aren't, and how you can use kind of styling to help get across some of that information. So yeah, that was an overview of some of the uses. Yeah, to end that bit, I just want to say, like, you're free to use these standards. This is a community standard. So yeah, feel free to go onto the SBOL standard website, download the symbols, use them in your talks, papers, wherever you like. If you have any questions about the standard, I'll show you so there's obviously the website has information on here, but there's also information on here about proposing new features and glyphs that you can go into. And there's also a link, let me try and find it. If we go to here, there's a associated repository which we actually use for managing the standard. And so in here you can actually raise changes to the standard, which is really valuable. So for example, you might be studying a bit of biology that not many people in synthetic biology have before. And so there are standard ways that you often represent maybe the certain types of parts or interactions that you're particularly interested in. And so this is a way of raising, kind of putting forward a proposal for new glyphs that you might want to see introduced into the standard. And those get processed by editors in the standard regularly, and then there's votes to see whether they get included or not, and then they get actually incorporated into standard as a final bit. So feel free to kind of yeah, interact with the community as you wish. It's a really, like, I have to say, I joined quite late on, I guess. SBOL being established for quite a few years before I kind of got involved. And it's a really nice bunch of people, and the community is really receptive to kind of expanding this and trying to make it as useful as it possibly can be. And I think if you look now at a lot of the papers that come out in journals, as opposed to say 10 years ago, you really start to see the kind of, these efforts have really started to pay off, and that it's much easier to take diagrams now from papers and understand what the author's trying to show than it was, say, 10 years ago, where there was all sorts of things being used for different things. There's still a long way to go, I think, but I think it's really helpful for the field and helpful for research as well. Now, to end this bit, I just wanted to quickly talk about some of the future developments in terms of where the standard's going, and ways, if you're interested, if you'd like to get involved, if there are aspects that you might want to focus on. And so I know from discussions, there are a couple elements which I know people are starting to kind of think about how we might incorporate into these diagrams. One of them is around RNA-based parts. So you will probably have seen there's kind of a lot of interest in kind of RNA aptamers and RNA-based circuits and interactions. There's historically has been quite a lot of work around kind of DNA-based computing and strand displacement circuits. And so we're starting to think about how we can perhaps incorporate elements of that into these diagrams to really have a more complete representation of functionally what's going on in these systems. And there is already some effort around trying to create what's been called kind of a protein design language. So looking at domains of actually, say, a coding region, and actually seeing how you might represent that to actually show, yeah, to convey visually what that protein, how it's structured in different aspects of that structure. And obviously there are, one of the things that the SBOL visual doesn't try and do is reinvent the wheel. So if there are good standards for representing these things, it might be that it just allows those representations to be used within it. But in some cases, it might also be useful to actually extend the standard to accommodate certain aspects of that as well. So it's very open to being used in different ways. And the final thing I wanted to highlight, because this is what, obviously you don't have to hang around for the development part, but the second part of this will be looking at some of the kind of actually coding some of these things and where that's actually going. And one of those areas in particular is around trying to move away from having manually specified glyphs within the standard definition itself. So historically on this repository that we use for actually managing the standard, the glyphs themselves have been created manually in Inkscape or Adobe Illustrator or whatever tool it is. Now that's fine when you've got a small set of glyphs, but as you get more and more of them, it can become problematic. And the reason for that is that the standard doesn't just specify that the glyph should look like this, it also specifies ways that that glyph, the shape can be modified and it still captures the same essence of that glyph. And those modifications or geometric customizations are impossible to capture computationally in just some free text that's in the standard itself. And so one of the things we've been looking to do is to make use of a kind of extension of the scalable vector graphics format that allows for parameters to be used within the definitions of the shapes within them to actually enable parametric versions of glyphs that can be then geometrically customized. And I can show what that means later on in terms of things, but fundamentally it means that if you've got, say, a CDS shape like this, it allows you to specify that actually when you extend that shape, you don't want to just stretch all of the shape itself, but you want it to extend the length of the CDS and keep this arrow at the end at the same proportion or whatever. And so there are ways of using parametric SVG to actually allow you to specify some of these constraints on the geometry, which is really, really valuable. And that's what we'll talk about a little bit in the next bit, or James will, I've got a little talk from James that I can present. So that was the kind of the end of the first bit. Are there any other questions? Okay, I can see a few in here actually. So can you represent phenotypes, e.g. expressing this protein leads to motility? So there's nothing, so the SBOL visual doesn't have anything related to phenotype per se, but it doesn't mean you couldn't use your own, I don't know, symbol or shape or image to kind of represent that. And I suspect, I could imagine, for example, especially in terms of motility, you might have your cell, you might have your circuit within your cell, and it might be that the output actually connects to, I don't know, if it's a bacteria, something like a flagella, and that would kind of represent that there's some kind of connection with that element. But there's nothing in the standard related specifically to phenotype other than processes like this. There's a process which maybe leads to transcription of this or translation of that, and it would be a process that then occurs which would lead to some motility in some way. But how you represent that is really up to you. There's no constraints on that. Cool, I got a thumbs up for that, good. Is there a specific locus nomenclature that you choose? Again, no. So the only, so I guess you're kind of referring to this where we're talking about positions in chromosomes and things. The only thing we're really specifying in the standard is that this means that it's located in the chromosome somewhere, and how you annotate this in terms of textually is up to you. And to be honest, I'm not an expert in all these different fields, but I know that the way people represent chromosomal locations is different across fields anyway. So it's probably, this would be one of the areas where we wouldn't be able to enforce a particular standard, because it might be that in your specific field, you have a very well-defined way that you do that, which is different from someone else. And so we're not instilling any fixed way that has to be done, but you can obviously annotate this with text however you like. And so that gives you the freedom to use the most appropriate method. Okay, any other questions on that? No, doesn't look like it. Okay. Cool. Another thumb. Actually, it's really helpful to have the thumbs up, because otherwise I don't know if that was helpful or not. Good. So yeah, so I guess those questions are highlighted. So there's a lot of things that aren't captured by SBOL Visual, and that's on purpose. It's not meant to capture everything you might want to do. We just want to help you more precisely show certain elements of your genetic or biological design, and we give you the freedom then to kind of use that in different ways to like, yeah, express phenotypes or to show elements about where these things are encoded. Right. So that was the first part of the talk. The second part, originally James would be here and he'd be able to give this talk, but unfortunately he's not here. So I have somewhere a movie that I'm going to play. So I'm going to put this full screen and then I'll play this, and then I can take any questions you might have about it afterwards. But it will highlight some of the reasons that we're now moving to, or trying to promote this use of these parametric definitions for glyphs. And I'm really closely involved with this. So if this is something of interest to you, then come and have a chat to me, because we're really trying to find people to sort of help us push this forward. So let me make this full screen, and then I will click. Actually, I don't know if I can make it full screen, but I can do this. Is this visible to everyone? Is that big enough? Yeah.
Yeah.
Hello.
Cool.
I'm going to talk about the use of parametric SVG to represent SBOL visual glyphs. I'm pre-recording this talk as I'll be away from home during the workshop, and I don't know whether or not my internet connection will be good enough to present smoothly. You don't need to understand what I'm about to say in order to draw SBOL visual diagrams. The details might be of interest to anyone thinking about creating new tools or about adding new glyphs to standard. As Thomas explained, the number of glyphs defined in the SBOL standard is now reasonably large and is continuing to increase as the standard broadens in scope. We also hope that the number of tools supporting SBOL Visual will continue to increase as the standard becomes more widely adopted. There's already an SBOL Visual ontology server, which returns details of the glyph matching an SBO term provided as a query. So you can tell the server that you have a promoter, and it will return details of the corresponding glyph. This can provide a way to lift knowledge of the glyphs from separate hard-coded representations inside individual tools to a common representation shared between tools. It could also provide a mechanism to deliver new glyphs to existing tools. However, simply providing static image files is not enough, as some tool authors have ended up having to write separate functions to draw each glyph in order to enable specific customizations. For example, a coding sequence is represented by an arrow, and it's common to want to scale the body of this arrow whilst keeping the arrow head the same size. This writing of functions is inefficient, and it creates an increasing maintenance burden as new glyphs need to be separately added to all existing tools. This also makes it harder to create new tools. What we really want to do is take the idea of a function that draws a glyph and find some way of expressing this in a portable way that can be used from any programming language. Our solution is a transition to using parametric SVG to define glyph shapes. Before introducing parametric SVG, I will briefly explain what SVG is. SVG is an abbreviation of Scalable Vector Graphics. It is a standard for 2D vector graphics that's widely supported by web browsers, as well as by various vector graphics editors like Illustrator or Inkscape. Vector graphics means that it defines an image in terms of its constituent geometric components, such as lines and shapes, rather than by defining the color of each individual pixel in a grid. This enables the graphic to be scaled up without becoming blurry. The standard is based on XML, which means it consists of tags or elements which are written between angle brackets. A tag can be self-closing if it ends with a slash. Alternatively, a tag can be closed by a matching tag that starts with a slash. Elements can be nested, so if an element is not self-closing, it can contain other elements. Elements also have attributes. The SVG specification is quite large and complicated, as it's expanded to include complicated features like filters for doing image processing. However, the core of the standard is still quite simple. There are a number of elements representing basic shapes like lines, rectangles, circles, and ellipses. There are group elements, written as G, that allow you to group together several other elements and do things like apply the same transformation to all of them, for example, to translate them all horizontally and or vertically. SVG also supports paths, which are curves that may optionally be closed. These are defined by a data string stored in the D attribute. The idea is like Lego or Turtle graphics. The path is defined by a sequence of commands, each of which defines one section of it. There are commands to move to a location without drawing a line, to draw a line from the current location to a new location, to draw a Bezier or elliptical curve to a new location, or to close the curve by drawing from the current location back to the starting location. Each command is a single letter followed by coordinates. So for example, this SVG here contains a circle element, a line, a path, and a group that contains one rectangle and a text element. The path has a data string that says to move, so the coordinates 150, 0, then draw a straight line from there to 75, 200, then draw a straight line from there to 225, 200, and then to close the curve. Parametric SVG is an extension to SVG that provides an alternative to assigning specific numerical values to attributes. It lets us instead assign a formula or arithmetic expression that can include parameters. We can provide default values for these parameters, but a tool can override these, e.g. to change the array length of a CDS glyph. The way that this works is by defining a new namespace called parametric, and then adding extra attributes in this namespace. So, as well as defining the conventional X attribute, we can also set parametric X attribute to be a formula written inside curly brackets. Most tools don't know what these new attributes mean, so they just ignore them and use the non-parametric attributes. They don't throw an error because adding extra attributes in a different namespace is perfectly legal XML. Indeed, if you export an SVG from a vector graphics editor like Inkscape, you'll typically see that it has added attributes in its own namespace that only it knows how to interpret. However, tools that do know about this parametric convention can assign different values to the parameters, substitute these into the formulae specified by the parametric attributes, and update the corresponding non-parametric attributes accordingly. This is not a widely adopted standard. It was proposed a few years ago by shape.cc, which I think was a company that no longer exists, and it was then apparently abandoned. I'm not sure whether anyone else is actually using it. However, we think that it is ideally suited to the problems facing SBOL Visual that I've described, so we're reviving it. We've created some of our own tooling around it, which I will talk about later. So here's a concrete example of how an old glyph specification image has been converted from a traditional SVG to a parametric SVG. There are a couple of things I want to point out. The first is that we use the class attribute to record what each attribute actually represents. As well as the glyph itself, we have a bounding box and a baseline, each with their own class name. These are important to include in the specification document as they show how the glyph should be used, but they are not drawn when the glyph is actually used in the diagram. The actual glyph shape is defined by the path element with the name filled path or unfilled path. As you can see, the parametric definition of the glyph paths are quite long and include many parameters. When the parametric SVG is rendered, each of these individual terms enclosed in curly brackets is replaced by a number. We've standardized on using the two class names of filled path and unfilled path, so that fills can be correctly applied to glyphs containing holes, such as the insulator glyph, so that the paths that enclose the background get filled with the background color rather than the glyph color. For clarity, is not shown in this code example, but we do also set the style attributes on elements to provide default styling, so that if the file is viewed in, say, a web browser or vector graphics editor, it will appear as on the right with this dashed line styling around the bounding box. Drawing tools should normally override this so that the bounding box is hidden and the glyphs are colored as specified by the user. Using parametric SVG files as the definitive definition of glyphs has advantages not only when creating tools, but also when developing the specification. Currently, the SBOL visual standard is defined in a Github repository, and when the standard is updated, GitHub Actions automatically performs several steps. These include exporting individual images of each glyph, both with and without the bounding box and baseline line, in several popular image formats, including SVG, PNG, and PDF. A palette or quick reference card showing all of the glyphs side by side is also generated. Integrating parametric SVG support has proved to be fairly straightforward, and we've developed a number of tools that work with it. I've written a React component that takes a parametric SVG file and an object of parameter values and renders this. This component is used by SBOLv, a JavaScript library for rendering SBOL visual diagrams in the browser. Currently, I think this uses a set of glyphs with a slightly different parameterization, but this will be harmonized in future. I've also written a command line tool in Python that reads parametric SVG files using libxml2 and then processes them in various ways. This can do things like exporting clean, non-parametric SVG files, producing a table listing the parameters used in each file in the directory, or performing substitutions to change how glyphs are parameterized. It was written to help SBOL editors developing specification, but it may be useful more widely. SBOLv is a Python library that can draw a parametric SVG file within a matplotlib plot. I think Tom's plan is to update his DNA Plotlib library to use SBOLv as the underlying renderer. So, to conclude, we think that parametric SVG is a promising way to define glyphs in a way that allows them to be programmatically manipulated. A collection of parametric SVG files has been created for existing glyphs, and support for them is being added to a number of tools. We plan to soon introduce a SEP that defines how parametric SVG files should be created for newly added glyphs. Thank you for listening.
Cool. So hopefully that gave you a bit of a idea of what parametric SVG files look like and what they do. And we said it's the direction that we really want to push the definition of the standard in, because it really helps us automate not just creating or like documents that are necessary for the standard, but also when you're developing tools, it means that you can then just hook into this kind of machine-readable format of the symbols, and basically it removes any need for you to write kind of individual rendering code for every single glyph. So it's like a really valuable way of doing it. Did anyone have any questions on on James's talk. I can try and answer them as best I can. Don't see any questions in the chat. So the last bit, so James mentioned about this SBOLv library. So this is something that myself, James, and a student of mine have kind of developed over the last year or so to try and really simplify rendering SBOL visual diagrams from Python code. And this is it's designed to be a very low level, simple library that pretty much anyone, if you can write Python code and you can create plots in Matplotlib, you can create SBOL visual diagrams using it. And so for the final part of this kind of workshop, I just wanted to show you some examples of how that looks. Do you see, I've got a question here, do you see SBOLv as the new DNA plot lib? So not really. So SBOLv is meant to be as small as possible. So basically it allows us to forget about how we render glyphs because it handles all of that. And so the idea at the moment is for this SBOLv to become effectively the renderer that DNA plot lib plugs into. And DNA plot lib can then focus more heavily on other types of rendering tasks. So for example, one of the areas I'm really interested to pushing into is how you render, for example, very large and complex genetic designs. So kind of how you lay them out, how you might abstract certain things, all of those sorts of elements, which aren't specifically related to the symbols you're using, but they're bigger questions that have to be addressed. And so I think DNA plot lib will become more, it will be thinking about bigger problems than just the rendering of the individual symbols, and SBOLv will really be that effectively a rendering library that it can then hook into. We really want to keep it simple and small so that other libraries can be built off of it. We don't want it to be a kind of something that is it is kind of a massive thing in its own right. It should be very simple so that we can manage it and maintain it quickly. So okay, so to show you how it works, well, I can show you there is a GitHub repository that you can go onto to actually have a look at it. So if you want to have a look, maybe Prashant, someone's quicker than me in this, but there is you can go to this link here. And so this is the current release. And we also have a paper that was recently put out on in synthetic biology from Oxford University Press. So that explains kind of what the toolkit is. And basically it's a very simple library. We don't have any kind of proper setup scripts for at the moment. So if you want to use it in your tool, you merely need to include this directory once you've cloned it in your Python path. And what you'll see is literally, it's effectively a single Python file. It's not even that long, but this basically handles all of the reading of the parametric definitions of the glyphs and the rendering. And within the package itself, we have versions of all of the parametric converted symbols that we can then use. And perhaps actually it might be interesting. I can show you, I don't know. So for example, let's get one that CDS. Can I look at the raw text? SVG. Okay. So stop. If I put this into an editor. Yeah. Is this big enough? Can people read that?
Yeah, it's okay.
Yeah, good. Okay, I can make it a little bit bigger actually. So you can see when James was talking about these kind of parametric definitions, these are standard SVG files, but they also have these effectively these additional attributes that start with parametric, and these are the elements that the library is able to take and then effectively calculate what the value should be on the fly. And so that allows us to then parameterize how long a CDS is or how wide it is, and it will then everything else will just update really, really nicely. And we have versions of these parametric SVGs for all of the elements in the current SBOL Visual 3 standard. So it's really it means you've immediately got access to all of the symbols themselves. Right, so that's where you can find it. And on here as well, I'll highlight, I'm going to go through some of these examples now, but there's a whole set of examples in a gallery. So we show how you can plot kind of all of the different symbols, how you can customize different aspects of it. I'm going to talk you through some of the things: animations, rotating things, labeling things, drawing interactions, and I'll talk you through those. But this gallery has more than what I will go into in this very brief kind of overview. So to get started, I will close this down. We don't need this anymore. We probably don't need that anymore. Yeah, I've shown you some of these parametric glyphs, and what I'll do now is I'll load up one of these first examples. So this is a kind of what I would call the 101, and if you want to use this library, how you would get started. So yeah, first of all, as I said, you have to have this SBOLv folder in your Python path for it to find the library. And then once you have that, it allows you to then import SBOLv, and you can then use it within your code. Now, the key object within the library is what we call a Glyph Renderer. And so what this will do is, what this line here does is it creates a Glyph Renderer object that basically reads in all of the internal parametric versions of the glyphs that are present within the library. And at the moment, that's everything in the SBOL3 standard. So this produces an object that we can then use in many different ways to render a particular symbol that we're interested in. And because this is a Python-based tool, to make it easier for us to be able to render and save these things and integrate them with other sorts of plots, we use literally Matplotlib for doing all of the rendering of our diagrams themselves. So in order to create an image, we need to actually create a figure and an axis on which to draw within. And so what this line here does, it just creates a figure using effectively Matplotlib. It then creates an axis that we can then draw to. And one of the things to be aware of is it's important when you create this Matplotlib kind of context to ensure that you have an even aspect ratio. So in Matplotlib, you can have axes that are not a one-to-one scale. They can be stretched in different directions. And so that can obviously make the rendering of these symbols look strange. And so it's really important to ensure that you have either the right aspect ratio just by the way you set it up, or you enforce that when you're actually creating the axis itself. And then it's really, really simple to use a library. You literally take the renderer, and then we have a function that allows us to then draw a glyph. And this draw glyph function basically takes an axis that it's going to draw to. It takes a name of the actual symbol that you're going to draw. And so this basically acts as a key into that library that I told you about. And then a tuple, which is just the position that you want that particular glyph to be drawn at. So this is going to, in this case, draw 3 different glyphs: a CDS, a promoter, a ribosome entry site, which is basically an RBS, you would have heard that term before, and a terminator. We then, in this case, just set the size or scale of the axis so that it's going to be drawn appropriately, and then we just show it. And so I can run this piece of code. What it will do is it will create this Matplotlib figure, as you can see, and then it will draw these elements, and you can actually see the axis there. I haven't hidden it, so you can actually see where it's actually drawing these things. But it will just render them to a plot. And so it's, like I said, this is really the simplest thing you can do. But it kind of shows you that you've now got immediate access to pretty much all of the kind of built-in symbols that are available. You can now render them in your Python scripts. Now, that's kind of like the 101. If you want to do something a bit more complicated, then we can create this figure, and we can actually think, well, actually, we want to customize some of the features or the parameters in these glyphs, or maybe style them a little bit differently. And so to do any styling or alter the geometry of a symbol, we have what we call user parameters or style parameters. Now, user parameters are basically just a dictionary in which we have a set of keys which correspond to the parameters within the parametric definitions of the glyphs. So if we go into, in this case, we're rendering a CDS and we're using these parameters here. So if I go into the CDS, what you'll find is it has some default parameters defined up here. And you can see there's one called arrow body height and arrow body width. And these are the parameters that we're actually messing about with when we put it in this dictionary. And we just send that to this rendering code, and it will then render that. If I comment these out first. Okay. And then I run that, what you'll see is what that does. So still, I did not save it. That's right. So you can just see the one that we're actually looking at. Okay, so this is this bit of code here that we're using here. And so what we can do is we can now use these parameters to alter the geometry of the shape. So we can, for example, maybe we want to make it longer. So let's make the arrow body width, let's make it twice as long. So now when I run this, what you'll find is, you probably can't see that very well, but there's actually 100 wide now. Make it, say, 500 might be more evident. Oh, have I done this right? What have I changed? Oh, it should be, I think it's width. Let's do that. It's going to be massive now. Okay, there we go. So this is a way of kind of messing around with kind of the geometry of the shape you've got there. I've gone way too big. Go big, go, there we go. So you can see that you can start to change the geometry of this. We want to change maybe the width of this height, this head, we can change that. So maybe make that a bit longer, make it a bit more pointy. So there's, and we can do this for pretty much any of the glyphs that are defined. And if you look in the standard, you can see what parameters are available for each of these symbols that you can start messing around with. So that's one way of changing the geometry of the things you're drawing. Now, another thing that you might want to do is actually alter the style of the thing that you're rendering. And so to do that, we also use a dictionary. But in this case, because you can have multiple elements making up your glyph, this dictionary has a key, which is the particular element that you want to edit. And then within that, you have another dictionary, which then defines the key value pairs of the styling attributes that you want to change. So in this case, we've got we want to render a CDS, and we want to alter certain aspects of the CDS element. And so if I look in here, what you'll find is that there's a path in here called CDS. And so this styling element, CDS, is going to have this style applied to it. So in this case, we're going to change the face color to RG, to blue, and we're going to change the edge color to something else, and we're going to make it a really thick line width. And similarly to the user parameters, you send these styling parameters in through just a user style option when you actually call the function. I'm also showing here there are some other functions, attributes you can use that allow you to kind of rotate where these change the rotation of the symbol, which is sometimes useful if you need to plot them on an angle or something like that. But if I've saved that, if I run this now, what you'll see is, so this is now plotting a blue arrow with a yellow outside on this specific angle, basically. And so you can very easily use this to automate the creation of quite complex diagrams using these symbols and style them in specific ways that might be useful for maybe conveying some information about that particular element in a design. So that's kind of some of the basic things you can do. What's the next one I've got on here? Another aspect that I mentioned that's really valuable when you're using coding is that you can actually create dynamic animations of genetic designs, which is perhaps useful. And so this is just an illustration of how we've integrated changing certain parameters, in this case of a coding region, so the shape of it and the color of it, randomly as this animation takes place. And so what happens is, probably easier to show you than to describe it, is it will basically generate an animation where at each time step it renders a CDS, but it randomly picks, for example, the different styling attributes, the length, the arrowhead width and height, and all these sorts of things. And so it's basically a demonstration of kind of the range of different styles that a CDS might potentially take. But you can see that this sort of approach can be used then for creating some dynamic animations or visualizations of the systems that you might be interested in. And it's really really simple to do. This is because these are Matplotlib axes that you're drawing to. You can pretty much do anything that you can do in Matplotlib. You can use SBOL visual symbols for. So that's one thing around animation. Oh, wait a minute. Got a question here. Was there a reason for differentiating the case of the key CDS versus CDS? No, it was merely one person used uppercase, one person used lowercase, and they'll likely be, as kind of James highlighted, probably a harmonization process by which we end up standardizing on one particular case for some of these things. Realistically, I suspect path names should be correct, like uppercase, if that's actually what they are, but that likely will change as this becomes kind of more stabilized in some sense. Okay, so that's kind of animated stuff. Now, everything I've shown you so far is just literally rendering individual glyphs, but to kind of give you an idea of how you can actually potentially render a number of glyphs that make up your design, we also include a construct type, effectively, or an object that allows you to basically specify a sequence of parts, and SBOLv will then render those for you, so you don't have to go through a loop and actually render each of those separately. And this is just a script to show kind of how you can set that up. So one of the ways that you can actually tell SBOLv what this construct consists of is just a list of parts. And so we have a list here, which we call part list, and we also define a name tuple called part, which allows us to capture then the fact that we need to define the glyph type, its orientation, and then these styling or geometric parameters that are present. And we can then literally just append a number of parts to this part list. So we've got a CDS, which is in the forward direction. It doesn't have any user parameters, but it has this styling. We'll then have another CDS in a forward direction. Then we'll have a promoter. And so you can see, you can very quickly just add together all of the components that you want to be shown. Again, you create this renderer. This is kind of used. This is always used by any of the functions within the tool. And then you can create a construct which basically is another object which takes this part list and this glyph renderer, and then it can actually be drawn to basically an axis. And so when we run this, what will happen, which version is this? It will take that list of parts up here, this thing here, and it will then just render them as a list of parts. So there you go. So you can see it here to the axis itself. And so this is kind of we added this because it just makes it easier for people that often you just need to render small segments of designs, and this allows you to very quickly create those objects and then just let the library do all the rendering aspects for you, which is really handy. So that's that. Obviously, once you can do something like that, it's really trivial to then extend it to take in as input perhaps some of these standard formats that people use for describing annotated sequences. So an example is what's known as a Gff format. And so this is a text-based format which is often used for annotations of genomes. And so you have this kind of, I'm not sure it's space or tab delimited format where each of these columns corresponds to some useful information. And so this is effectively the chromosome. I can't remember what this column's for. This defines the type of the annotation. Then you have kind of a start and end position, some other information. There's the orientation, effectively the strand that this annotation's on. And you can have some extra attributes as well. And so I guess all I'm trying to say is that the nice thing is that you can, because the rendering is handled by this library, you can say, well, okay, I want to render this specific design. I want to render this GFF file. You can very quickly write some simple processing code. So I'm not going to go through this, don't worry, but basically some parsing code that can take one of these files and extract out each of these elements and generate a construct that actually encodes those various elements in the right order, and then just render them immediately to the screen. And so that's what this example does. It takes this specific GFF file and then it's able to render this particular design. And the really nice thing here is that this code is not specific for that design. We can give it a completely different GFF file. So if I, for example, remove all of these promoters and then I rerun that, what you'll find is this won't then have any promoters. It's very generic. You can very quickly use this for rendering all sorts of different sorts of things that you might be interested in. And I think this type of this ability to kind of now forget about how you do the visualization and just focus on getting the data in the right format that you can then just throw to the library for it to actually render it for you can really accelerate you actually creating complex diagrams automatically from kind of data that you might have that might represent the designs that you're actually exploring. And then the final example I wanted to show you was just something, kind of a tool that we created from, which was to try and replicate. So historically, there'd been a tool called Pigeon, which would allow you at the command line to kind of type in some plain text a construct, and it would then generate a visualization of that construct. And this is really useful when, for example, if you're making a presentation, you might just want to draw a simple little thing, and it allows you to do that very, very quickly. And so one of my students, Charlie, in not very many lines, if I'm honest, there's some, how big is this file? It's a couple hundred lines, and a lot of this is comments, if I'm honest. He created a command line tool that can actually parse text that's in a specific format and create a design from it. So to show you what this looks like, I've got a script here. So I'm just going to run this first so you can see what comes out the other end, first of all. Okay, so this has just created this thing here. So you can see this is actually, this is quite a complicated design. It's got some interactions and it's got different colored things and there's all sorts of stuff going on. But the way that this has been, the way that the tool's known what to render is just through a string of text. Can I wrap this? Here we are. So you can see that we have this command line tool. We provide a string, which is this bit here, and this defines the sequence of genetic elements on our backbone. And then we also define an interaction set, which defines which pairs of parts have interactions between them and what sort of interaction they are. And then we just define where to output that particular thing to. And the really nice thing here is that we have a very simple syntax where P means basically promoter. We want that one to be black. R means RBS, we want that one to be white, C means CDS, T means terminator. And so you can very quickly type out what you want for a design and it will then effectively render that to a file for you. So if we wanted to change this a little bit, maybe we want to take out this spacing element here. so this is the spacer here. If I just remove that, we can then just run this again. And this will then not have that spacer element. So you can see that's now being removed here. And so you can use this quite interactively to do that kind of stuff. And all that the only reason I wanted to show you this was that it's incredibly quick to make little tools like this using this library. And one of the reasons we want to keep the library small is that we don't want to make some massive tool that covers every use case. We'd rather you just wrote a little bit of code to use this in a way that is most suitable for your use case. And so the hope is that this can really just be used as kind of a rendering backend that completely removes the need for you to worry about how do I draw a CDS or how do I draw this, and if the standard expands, how am I going to accommodate that? You don't need to worry about that. The library will handle all those things. You just need to worry about what is it I want my specific tool to do with that capability. And so that's one of the goals of the tool. And I think it's yeah, I think it'll help support a lot more tool development sort of like going forward. I've been talking a lot. Are there any questions? Oh, there's a few questions on this. Okay. So I've got one from Michael. If you were to speculate on a stack to use to display a Parametric rendered diagram for a GenBank file, what would you use? A stack to use? So I have actually, if you were to, so the question's asking about, I think, and you can correct me if I'm wrong, how we might go about rendering, for example, a GenBank file rather than a GFF file. And I thought I had one on here. Maybe I don't. Oh, I do have one here. So we actually, that's one of the examples that we have on here. So for those who don't know, GenBank is a kind of a typical format that's used. Oh, this is quite a big, I can download it though. It's a typical format that's used for, again, annotations of genomes. And it's a little bit rich, well, it's quite a bit richer than the GFF format. And it allows you to kind of say where certain things are, as well as actually including sequences and other sorts of information. And so in terms of the question, we have, it's very easy to basically parse that file and pull out whatever information is relevant for you. So there isn't really the need to build a stack per se. It's more just a parser that can take a GenBank file and generate a construct with the right elements within it, as well as maybe styling options that you might be interested in. So as I said, this is a script. You can have a look on the GitHub repository to see how it works, but this is able to basically read in a GenBank file. And here we've basically specified kind of a very simple mapping from the type of element in the GenBank file to a color of the element in the visualization. So we're showing certain types of element that we want to render. And so when we run that on a genome, we're able to then basically show the genome with these little elements colored depending on the function of that particular part, effectively, from the genome. Obviously, the GenBank file encodes a lot more information that you could parse and represent in different ways, but that's really up to you in terms of how you'd want to actually do that. Oh, good. I see the example directly answer that. Good. So yeah, I guess the point I want to make is it's really easy to do all this stuff, and that's what I think would be, we'd like it to really act as a very simple library to get people to start using these symbols more widely and to automate the development of their diagrams and things. It really does save people a lot of time, and it will make the diagrams a lot crisper as well, because a lot of this is vector based, so you can scale it to all sorts of sizes. Okay, does the GFF input allow display variants? I'm not sure what that question means. Display variants on the
maybe I can just clarify. So yeah, just with the GFF input, I believe you should, within GFF, you should be able to specify mutants or variants. And then the question would be as to how you would get the para SBOLv to display a variant.
Yeah. So we just don't handle those elements at all at the minute. But you could realistically, it wouldn't be too difficult to process those and alter how things are rendered. I'm guessing if you're looking, say, at I don't know, like a protein library or something like that, you might have, you might want to render where deviations are across the CDS, and you might make the CDS the right length so that you can actually properly compare and see where mutations maybe are happening within it. Those aren't things that SBOLv is designed to do, but it's something that it wouldn't be difficult to do in terms of to implement as like an additional functionality beyond what it currently is able to do. I haven't gone obviously into any of the details here about how it works, and there's really nice documentation that Charlie put together with it, so it should be fairly easy to figure out what's going on. And something I didn't say about was when it draws things to the screen, it actually gives you bounding boxes and tells you where things are, so you can very easily kind of overlay information on top of the diagrams it produces. So it's kind of set up to be extended in lots of different ways, and that's really the hope that we have with it.
Yeah, I think that makes sense. And I guess then, so because I think I see all these possibilities, so I'm just asking, do you already have it? I guess the next one would be for multiple sequence alignment, right? So this is so while the SBOLv display is sort of a summary of the DNA sequence, you could imagine actually displaying an alignment of multiple sequences.
Definitely. And you could, so one of the things, we obviously don't do that at the minute, but we could, one of the things you could also do is, one of the reasons for introducing these parametric definitions is that, like I said, you can scale things appropriately. So you potentially can actually make a CDS the right length for how long it is in the genome, so that when you're looking at two CDSs, you know if one's bigger than the other, and you can actually compare them, which you really need to be able to do for example, sequence alignments and things. But what you can also do is ignore some of maybe you're not so interested in like base pair mutations, but you're more interested in whether certain parts are present or not present when you're aligning stuff. So you're not interested in the nucleotide alignments, but more kind of part type alignments. And that you could also do in this type of framework and fairly easily. I guess I don't want to, there's a lot of functionality missing, but the functionality it provides makes it very easy for you to not have to worry about the visualization bit. You just need to think about how do I want it to draw things and where. So there's that element sort of taken out of it. And really the key thing for us, so I developed this library called DNA Plot Lib for a number of years, and the real problem there was that every single glyph that's drawn has its own unique function that I've had to sit down and write, and that takes a lot of time. And when the standard changes, I'll go through this again and rewrite it, and it's just a real pain to maintain. This library doesn't, if there's new glyphs, we don't have to do anything. We just take in the new para SVG file, and we immediately can make use of that glyph and edit it in ways that are allowable. We can style it, we can do all sorts of stuff with it. So it's a real, it really helps remove a lot of the bottlenecks in terms of maintenance, which means if you're going to build off of this, you don't need to worry so much that, is Tom going to get around to adding that glyph that I really want kind of thing later on. You don't need to worry about it. You could create your own glyph, or you can just wait for it to be ratified and it automatically gets sucked up into the tooling. So it's really trying to remove a lot of the bottlenecks which have kind of hampered the standards. It's grown to a size which becomes kind of unmanageable doing things manually, if that makes sense.
Yes, and I think it's quite useful. I'm not seeing it as missing functionality, but rather as opportunities for your group or anyone else to develop on top of this foundational library. Definitely,
yeah, that's the hope. I come from a kind of a upbringing using a lot of Unix systems, and I like the methodology of you should have tools that are quite simple, but you can use them together with other tools very easily. And I see this as kind of really taking away the need to worry about how do I draw glyphs or worry about managing that, and just pull it into other tools that you're developing very, very quickly and easily. I'm hoping we've only recently released this, so we've been playing around with it for a while, and we use it internally in my lab for different things, but we're still at the stage of seeing, you know, who takes up these things. But to give you an idea, like the DNA Plot Lib libraries, it has like several hundred labs around the world using it for different things. So that there's definitely an appetite for using these tools. I'm really hopeful that all these kind of little solutions can be re-released so people can build on them. That's kind of the hope. But even if they're not, having a really robust kind of foundation to build these tools with is really valuable going forward, I think. Cool. Is there any other questions or comments? Yeah, I've got some love from Prashant there in terms of he likes DNA Plot Lib. A lot of blood, sweat, and tears went into making that easy to use, and it does produce some really nice diagrams. And I really think that once we can replace the rendering backend with this, it's going to make maintenance longer term much easier, and means we can focus on more interesting things. Like I said, maybe multi-chromosomal layouts or multicellular type stuff. We can do all that we can put effort into that rather than just maintaining glyph libraries, which is not the most fun thing to do. Okay, well, I'm happy to answer any other questions via email if people are interested, or yeah, through other discussions and stuff. Just let me know. And yeah, I hope that was useful, and it's given you a kind of an idea of what SBOL Visual is, some of the ways you can create these diagrams and stuff. But yeah, we'll probably best to, I think, end it there. So yeah, thanks for hanging around until the end.